Okay. All right. Everybody, please. This working? Can everybody hear me? Online as well. Need to talk through the microphone. All right. So hi, everybody, and welcome to the Function COSI 2024. My name is Ido Friedberg. I am organizing it, and together with Duca. Are you here? Okay. Say hi, Duca. And Ana Lojas. And Mark Loss. So any complaints, go to them or to me. And I'm very glad to welcome you here. We're going to have a fun 2 days together in the session. So I'm going to talk a little bit about ISMB 2024, ISMB in general, because some of you are new to this conference and are kind of confused by what the heck is going on here. What are these COSIs things? What is a function COSI? How did I get here? And so on and so forth. It's a little bit of a non-traditional kind of conference. So I'll talk about that. I'll talk a little bit about our activities as the function COSI, the communities that we work with, and hopefully those of you who are new to here will join us, and a note to the speakers as well. So if you are new to ISMB, and this is your first, how many here are, this is their first ISMB? All right. Okay. Well, okay. So ISMB is really the flagship meeting of the International Society for Computational Biology, ISCB. I think that most of you are members because it's cheaper to register to this conference as a member than not. If you're not, well, you're lost. ISMB has been described as a confederation of COSIs, communities. COSI stands for Community of Special Interest. And the idea is that we have all these COSIs that deal with microbiome, RNA, structural biology, high-frequency sequencing, translational medicine, machine learning, blah, blah, blah. All of these are around you, and you're welcome to go around and explore. They're all supposed to be, all the talks are supposed to be synchronized under 20 minutes. So, you know, if you see a really great talk, I want to go to see an RNA, you can go and see it there. Just, you know, try to leave unobtrusively. It's common and people do it. There are also special sessions that are not related exactly to COSI. We have an NIH data science session here, digital agriculture. There's a whole hour on demystifying scientific publishing, which I haven't been able to do in 25 years, but whatever, and so on and so forth. It's really a very busy conference in terms of the offering. So please remember to breathe and take breaks outside the conference. My first ISMBs, I was totally conferenced out by the third day because I was trying to ingest so much. I had such a big FOMO, and you know, you do want to take it easy. So you know, you will miss stuff, but that's fine. The important things actually happen in the halls, as we all know. What is the function COSI? What is the specific session you're standing in? So it was founded in 2005. We called it the AFP SIG, Special Interest Group. So SIG morphed into COSI, Automated Function Prediction morphed into function. So we're AFP and function COSI now. This is our 19th birthday. Yay, legal to drink. Our mission is to bring together computational biologists, biocurators, experimental biologists dealing with the important problem of gene and gene product function prediction, to share ideas and to collaborate. Legal to drink in Canada, not in the US, I should say. So what is function? What are we talking about? We're talking about function here. Anything and everything that happens through a protein. And we look also at how we do computational description of functions, usually through ontologies, but not only. We're actually very closely related to the ontologies group. We sometimes have joint session with them. So that's kind of the overlaps that go on here at ISMB. Go captures molecular function, biological process, cellular component. You'll hear a lot more about that later. Human phenotype ontology, capture phenotype in diseases. And our keynote speaker will talk about the uses of the EC, some of the ideas that behind the use of the EC that captures enzymatic function. Another dimension of function that we work on is identifying functional residues. So all of these things sort of go together. And we have two main activities. One is the annual meetings at ISMB, in which you are. The other is CAFA, the Critical Assessment of Function Annotation, which I'm going to talk about a little bit here, which is a computational challenge. As you probably know, a lot of ML methods go through computational challenges, which help assess algorithms to predict the function of proteins in this case. CAFA is a really time-constrained computational challenge. This is an example from CAFA, from the third round of CAFA that we did, but it's all pretty much the same. We released targets of proteins of which functions are unknowns. We asked people during the prediction timeline that you see here between t-1 and t0 to predict them. At t0, we said no more predictions, all bets are off. And we wait for a while for some of these proteins to gain function through, literature, biocurations, and experiments, and so on. And we do, after 7 months or so, a preliminary benchmark collection. The percentage is very small, but we have a very large data set, like 100,000 targets overall. So about 3,000 of those, about 3% collect functions, and on those we can do the assessments. And we do a final benchmark correction about a year later, and then we publish a paper. So everybody who predicts gets together to do that. And CAFA over the years has evolved a bit. We had 5 rounds so far. These are, the first CAFA was run in 2010. We had 59 models and 30 teams participating. Then it started growing and growing and growing until CAFA 4 had 148 models submitted by 70 teams. And then in CAFA 5, we had 28,000 models submitted by, this is actually wrong, not 1625 teams. We had 1987 teams at the count count. So why this jump in, suddenly the popularity of CAFA? Well, the reason is money, of course. Kaggle, a platform that does these kind of predictions, approached us and say, hey, how would you like to do some money to run CAFA? And we said, yeah, okay, we'll do that. And $50,000, and they gave $50,000 in prize money, and gave us a programmer and resources, and basically had CAFA run on the Kaggle platform. It was incredibly popular. We learned a lot. You're going to hear a lot more about CAFA 5 this afternoon from Clara, who in Predrag Radivojevich's lab will talk about the findings that we had in this competition and the results. You're also going to hear 5 or 6 talks from the top performers in the CAFA Kaggle competition, or the CAFA 5 round. And we're looking forward to a very, very exciting afternoon on that. So like I said, more about CAFA in general and CAFA 5 specifically today after lunch. It's really become an incredible crowdsourcing kind of thing, and we have some really fantastic findings. So please join us after lunch. A bit about the meetings that we are holding. The first Function COSI meeting was held in 2005 in Detroit. Almost annually since, including 2 annual meetings we had during COVID. We tried to have as much support as we can. We were supported by, in the past, by NIH, DOE, the National Science Foundation, Elixir, IEEE. We spent about $45,000 in student travel fellowships. Right now, we're in a bit of a funding gap. Hopefully, we'll get some funding soon, and we'll be able to resume some more funded activities. So that's kind of a little bit on the history of the Function COSI meetings. And what we really tried to do here is to bring communities together to work. And those would be experimentalists, computational biologists, and biocurators all to work together. This comes together very nicely in CAFA. For example, in CAFA 4, we had experimentalists providing us with data, and us providing experimentalists with testable hypotheses. And we published on that as well. Computational biologists, of course, most of you here are that, so you know. Biocurators are those people who provide, capture, and sharpen the data towards what we need to do. So those are the people that everybody needs to talk together. And in today's keynote, Valérie de Crécy-Lagarde will talk a lot more about the importance of that and how we could do a lot better. And I take this not as an admonishment, but as something that would be incentive towards thinking and talking with people outside your kind of knowledge base. So our keynote speakers, I'm very happy to have them here. Valérie de Crécy-Lagarde from University of Florida will talk today. Tomorrow morning, Rafael Nachmanovich from the University of Montreal. Rafi is also the co-organizer of the 3D SIG, the structural biology SIG. Again, some of the overlap, structure, function, and you're welcome. You should go there as well and see what they are doing. It's a pretty awesome SIG. So what are we looking forward in these next 2 days? Today we're going to talk about duplicated genes and unknowns, a lot of following Valerie's talk, which will kind of be a little bit methodical on that. Overview of the CAFA5 results after lunch, and talks from the top performers in CAFA5, plus other things. This is kind of, it's hard to do full thematics in this rather broad field. And tomorrow, Rafael Nachmanovich will give his talk. We'll talk more about structure and functions, and we'll have a whole session on large language models and protein language models, unless we watch the game between Spain and England, one of these. So speakers, how many here are new speakers to ISMB? Some of you, okay. So please just remember to be on time, upload before the session starts. Anne here will help you. And stay in the time allotted. We are very aggressive moderators. Try to leave a couple of minutes for questions, and you'll be signaled by the moderators and how much time you have and whether they're going to throw you off the stage physically. A lot of people are doing a lot of things in this community, and there's a lot of people to thank here. So Predrag Radivojac, hello. Is here. He's the president of ISCB, but in his day job, he works on CAFA and his lab as well. So Clara, his student, will be talking more about that. Rashika and Yusuf from Pedja's lab were working on that as well. Parnal Joshi from my lab at Iowa State University. And regarding the partnership with Kaggle, we had a lot of help from the ISU Technology Transfer Office to seal the deal with Kaggle and work with them. So I'd like to thank them as well. From Kaggle, Dee Scully, the CEO of Kaggle, approached us initially, which was great and very flattering. Walter Reed was the programmer who worked on everything CAFA and tried to understand our convoluted mind and our assessment metrics, which were totally unorthodox as far as they were concerned. Maggie Dempton and Addison Howard did all the logistics and gave us the necessary resources to run CAFA5. I already introduced our meeting organizers, Anna, Mark, and Duca. I would also like to thank the ISMB staff for all the incredible work they did for this COSI and for the other COSIs. So Seth and Bell. Special thanks to Lucy Caldwell, who basically hooked us up with Kaggle. Lucy works at Google, and she's a big functionista and function predictor by herself. Sean Mooney, who basically keeps our website alive and our morale up. And to the 1,987 CAFA5 participants. So thank you very much all, and thank you all for attending this session. And I think I'm done. So if anybody has some general questions before I introduce Valerie, that would be great. Thank you.
Thank you so much for organizing all this, of course, because it's really tremendous work. I was wondering, though, why do we have to only talk about proteins for function?
So we don't. I was wondering if I could put another bullet point in, but I'm bad at Beamer. Actually, there are a couple of posters and works here that actually talk about RNA function. And what we need to do is a little bit of division of labor between ourselves and the RNA SIG, and I haven't talked with those guys yet. But I will. But there are a few RNA. We didn't get them to be as talks. By the way, we had a huge number of talks here, a huge number of abstracts to select from this year. So we're very flattered. And I'm sorry if some of you didn't make it to the talk thing. But there are definitely posters, including RNA posters, that are around. And we did not throw those posters. We did not put them as posters because they were RNA. And we think RNA is second rate. No, it was just a really hard problem to get everybody in. So it was, yeah. So you're right, RNA is something.